Okay, so basically I want to tell you, basically this is more like a storytelling. And what I have learned by analyzing the whole genome sequencing data, building the infrastructure, and also help the science, and using both statistics, machine learning, and AI. So maybe I give a quick overview of the timeline of the whole genome sequencing effort. And so about 20 years ago, that was the completion of the whole genome and sequencing that basically sequenced one person, very, very expensive. And then about 15 years ago, that was a completion of the 1,000 genome program, sequenced about a couple thousand people. It's still very expensive, only the depth is pretty low. In the last couple years, several large whole genome sequencing programs have been launched, both in the US and also in the world. And the most well-known one is called the Transomic Precision Medicine program, called the TopMed. We have sequenced about 190,000 people. And the other is the genome sequencing program by NIGI. We have sequenced about 350,000 people. And both those data are available and, in the cloud. And the TopMed are available in NHLBI Biodata Catalyst. This is NHLBI ecosystem. And the genome sequencing program is available in Anvil. So that is the NHGRI ecosystem. And also multiple biobank has been launched, including the most well-known one, UK Biobank, and just a couple weeks ago, and the UK Biobank has announced the 500,000 people whole genome sequencing data are available in the cloud. That is the RAP. And also, Million Veteran Program plan to sequence 1 million people. ALPHAS used to be called the Precision Medicine Program by Obama administration, and later on it changed called ALPHAS. And so the plan to enroll 1 million people, and also sequence them, integrate the genomic data, epidemiological data, and the electronic medical record. And so this is the current status of ALPHAS. And so the plan is to enroll 1 million people across the US. And so far has enrolled about 700,000 people, you can see across many different states. And so with electronic medical record and also biosamples. And so far have sequenced about 250,000 whole genome, and the data are on Anvil. And so the data all cannot be downloaded, all in the cloud. So give you a sense how big the data are. So this is the TopMed, a freeze 10, about 190,000 sample. Before the QC, there are about 1 billion variants. And so after the QC, there are about 855 million variants. So the data we deal with are very, very large. So the scalability is critically important. And as you look at the data, and so majority of the data are the rare variants. For example, 45% of the variants are singleton. That means among those 200,000 people, only one person has it. And 15% are doubletons, and then only less than 1% are common variants covered by the GWAS chip. And so what is the goal? The scientific goal is we want to scan the whole genome to identify the genetic variant set or the mass associated with the human diseases. We don't care whether the whole genome is associated with the diseases. That's not useful for drug discovery. And so we want to see a particular gene or particular mask and of the gene associated with the human diseases, and this can help with the drug discovery. So then the question is, how can we do that? So what are the challenges? First, how can we define the variant set? And so use this example. Suppose the outcomes, the station called outcome, and in genetics called phenotype, is LDL, this is bad cholesterol, low is bad, high is low is good, high is bad. And then we want to see how the variants, for example, in the ApoE enhancer, suppose there are 100 variants associated with LDL, we control for the covariates and X and then here, because if you want to do 100-degree freedom test, so basically there's very little power. But if I know there are only 4 signals, so really the cultural variance, and with beta not equal to 0, we want to do 4-degree freedom test. But this is impossible in practice, and this is completely oracle. And also the SNP are correlated. So the question is, how can we empower rare variance association test without doing this 100-degree freedom test? And so basically the main idea is, suppose you calculate the marginal association statistics of each SNP and the phenotype, we want to aggregate them across those 100 variants in the ApoE enhancer. But how can we aggregate them? So in order to do that, we have to build, first we need to build the infrastructure. And so the first, we need to build a whole genome sequencing integrated analytic ecosystem. And so the most statistician, we are not trained as a software engineer. But I want to make the point, it's worthwhile for statistician invest ourselves to build data and analytic infrastructure and also computer engineering. If we wait for others to develop those things for us, it's too late. And we are likely to make an impact. Okay. So to show you, so in order to do this analysis, we have a genetic variance and also the biological function of the variance and the phenotype statistician called outcome, like LDL. And first of all, we want to integrate the functions and in the genetic analysis, and basically if the variants had more biological function, we want to give them more weight. This will help with the discovery. And so the next part is we want to understand the causal pathway. And so basically this is where the causal inference come in by integrating the whole genome sequencing data with other type of functional data like multi-omic data. And then the endpoint is the action. So that includes the prevention intervention strategy and treatment target and the new drug discovery. And so this is optimal. So in order to do that and to do this whole pipeline, we need to do some homework in the infrastructure building. And so the first infrastructure building, I'm going to focus on it to do the top. I'm not going to talk about the bottom. And so how can we do the functional annotation? And to this end, it's important for statisticians to get involved in a large consortia. And so for the whole genome sequencing, for the genome sequencing program by NIGI, and we have this genome sequencing program annotation working group I chaired. And so the idea is to integrate multiple functional data from many different sources, and such as the ENCODE, roadmap, and so on, into this large database and GTAC. And so we include over 300 annotation, 80% are built on 38. This include allele frequency, conservation score, protein functions, epigenetic score, and many others. And so if one think about the whole genome, totally there are 3 billion positions. And each position, there are 3 alternate variants. So totally, this database has 9 billion variants. And so this is a huge database. And so basically, I don't know, I was never trained to be a software engineer. I never trained to do the database. So, but in order to do this, we have to build a relational database. And so this is the PostgreSQL. So I hired a computational biologist. I hired a software engineer. I know what I need, but I don't know the technical detail. I don't need to know the technical detail. They are much better than me. And build this whole relational database, and also with very efficient indexing, and can query it and to find a particular variance in less than 1 second. And also, so for this is publicly available, and one can do the query for a single variance or variance in a gene, variance in a region, or you can do batch annotation. And because if you look at the TopMed data has about 1 billion variants, so I need to pick from this 9 billion variants database, pick up this 1 billion variants. But in UK Biobank, and they have 1 billion variants as well. But UK Biobank, this 1 billion variants are different from the TopMed 1 billion variants. So basically, you have to do this type of annotation very, very quickly. And also, in order to understand this, and because many of the users, they don't have computational biology background, they don't have the really genetics background, so we have to make it useful and for the users. So what we did was we built this Fever GPT. And so basically, try to help the users and to harvest the power of a large language model to improve the user experience using the Fever, this 9 billion variants database. So this, you can see the software engineering generally involve backend and also front end. Backend is a database, and so what we have is PostgreSQL. Front end is basically how can you do the query. And so basically, we built the Favor GPT and using the Favor API, and which include the ChatGPT-4 API and also Llama 2 API. So basically, we integrate this 9 billion Favor variance database and the documentation with the large language model API, which include the ChatGPT-4 API and also Llama 2. Llama 2 is by Meta, and Llama 2 is open source and compared to ChatGPT-4. So that is easier when we build the APIs. And so basically, those are the first person, Tom Lee, is a high school student. And so it's really, those high school students are brilliant. And so we have new software engineer and computational biologists. And so we basically have built this Favor GPT by integrating this 9 billion variance functional annotation database and also with this front-end interface. And so using the large language model GPI, this can do very, very user-friendly query and also easier for people to understand. So for example, this is the front page of the Favor website. So basically, you type in your query, and so just say, even somebody knows nothing about genetics, you can say, provide the functional information of BRCA1, or provide functional information of particular variants, or ask how many pathogenic variants does BRCA1 have, and then that will calculate, BRCA1 has 15 pathogenic variants, just looking at that database. So this is really easy for people to use. You don't need to do any programming, and so this is really convenient. Okay, now we have everything those built up. And so the next thing is, how can we borrow this infrastructure into the analysis? And so what we do is, like, suppose you have the data from the TopMed data or UK Biobank data. And also we have this backend database, functional annotation database with 9 billion variants. And we make that available in all those, cloud ecosystem, like NHGRI is Anvil, and NHLBI about Data Catalyst, and also the UKB is RAP. So we'll work with all of them closely, because that is what you have to be in the club. You need to build a relationship. They build this kind of a trust. People trust you, then you can work with them. So for example, for the TopMed data, we have this annotated data in Data Catalyst offered to all TopMed investigators, and they use it. And that makes things much easier. So you have built this type of reputation. And now, for the whole genome sequencing data, the data generally are stored in VCF format. And so basically, first thing you need to do is to QC the data. And so my colleague at Broad Institute, the Ben News group, developed the Hill, and that can be used to QC the data. So Ben's group and I, we work closely together. And then the next step is, suppose in TopMed data is there 1 billion variants, and then we need to use this backend database to annotate this 1 billion variants. So basically, we build this Favor annotator, and that basically is a script that can be used to annotate this 1 billion variants in TopMed or in the UK Biobank. And also, VCF files are too big. These are not used for large biobank size whole genome sequencing data. So basically, we use the GDS format, and including the annotation, and the compression rate is over 1,000 times. So cloud are beautiful, but cloud is expensive. So the data storage is expensive. So if one reduce from a couple hundred terabytes data to 100 gigabytes data, that costs you nothing. Okay, and so one of my colleagues at Stanford and his lab members tried to use the cloud, Google Cloud, and they now realize it's so expensive. And then by the end, he couldn't have enough funding from the grant to pay the cloud cost. He had to use his discretionary fund and to make up the cloud cost. So therefore, scalability and cost efficiency is critically important to us, and to make the method used. Now we have all those infrastructure built, and then the analysis pipeline we call the STAR pipeline that can be used for whole genome sequencing analysis. And one might wonder how much it costs to annotate the genome with 1 billion variants. It turns out we can make it cost only $24 in 3 hours. So that costs nothing. So that is very important. Whatever we want people to use, it has to be scalable, it has to be computationally efficient and cost efficient as well. now we are ready. And so basically what we have been doing is to work with multiple NIH ecosystem and also the UK Biobank ecosystem. And so basically do a different way to do the data sharing. The old way to do data sharing is basically cluster model. So basically you copy the UK Biobank data, Tianxi make a copy, I make a copy, and both of us have 2 different copies, the same data, and that's really not cost efficient. If 100 people in the room, we make 100 copy to our own computational cluster. So basically that is data sharing is the same as data copy. That is not efficient way to sharing the data. And because whole genome sequencing data too expensive and too big. And so right now the change to the model is bring the researcher to the shared data through the cloud, also only 1 copy. For example, like UK Biobank whole genome sequencing data is in wrap by DNA nucleus, everybody access the same data. And in order to do that, one need to get familiar with workflow description language like WDL, and also need to be familiar with the workspace and the AppleEd. And also Anvil, the genome sequencing data are in Anvil. So and also AlphaS data are in Anvil as well. And so basically one need to get familiar with those kind of cloud ecosystem. And the TopMed data are in Biodata Catalyst. Gradually, the DB gap will disappear. And so this will be replaced by those NIH ecosystem. Okay, so the next point I want to make is empower real-branch association test by incorporating functional annotation. So basically the key point is to integrate the domain knowledge and in statistical inference. This will help increase the impact of statistics in science. So that basically means we need to learn, like in my field, we need to learn genetics. We need to have my student postdoc learn genetics in order to make the method really useful and make an impact. Okay, so how can we do that? And so this work was co-led by my former students. And also, the other thing is we need to publish in statistical journals. And so like the first paper was published in Nature Genetics. And the second paper, we want to make everything automated. And so we create a whole pipeline. And that was published in Nature Method last year. So the idea is to empower real-branch association test by dynamically upweighting the functional variance using multidimensional annotation score. So here is the pipeline. So basically, we use so first we create the phenotype and the covariates and also sparse GRM matrix, and also we annotate the data. And so as I mentioned, BCI file is not scalable for large-scale whole genome sequencing study. So basically, we use the GDS format. That compression rate is over 1,000 times. It's much, much cost-effective for dealing with the large data. For common variants, we can do the single SNP analysis. For rare variants, we can do either the gene-centric analysis or genetic region analysis using the coding mask or non-coding mask. And for the coding mask, one can group the variants. Basically, they all analysis, you have to know the biology. And so including so for the coding mask, we group the variants into loss functional variants, or the missense variants, or synonymous variants. And for the non-coding variants, we group them into enhancer and promoters. And also, one can do the sliding window approach. One challenge is when one analyzes non-coding variants, it generates functionality are tissue and cell specific. So the non-coding variants analysis is more challenging. And so to annotate the data, so basically, because there are many, many different annotations, for example, if one look at the protein function, there are over 10 protein functional score, and also for epigenetics from the ENCODE, there are over 30 epigenetic scores. So first, we need to do some dimension reduction. So what we do is we use a principal component. This we call the annotation PC. And for example, for the 30 epigenetic score, we calculate the first principal component. For the 10 conservation score, we calculate the first principal component, and so on. And then basically, when we talk about functionality, the variance functionality cannot be captured in a single dimension. So if you look at the heat map, the correlation about epigenetic function and also conservation score has very little correlation. So that's why we need to use multidimensional annotation. And so here is the analysis workflow. So we had input the data, and then we run the favorite annotator to annotate the top metadata. So create annotation score for each of the variants, use multi-dimension. There is a black screen. Is something broken? Yeah. Oh, really? Okay. Yeah, that's right. This basically is the dimension reduction or embedding. You can think about the embedding is a fancy way of saying dimension reduction, basically. Yeah. So then we create those kind of annotation. Now we can do group the variants into different masks, and so for each gene, we group the variants into loss functional variants, or the missense variants, or synonymous variants, or promoter and enhancer. And then we can run analysis and using a different weight function, and then we aggregate those weight functions, aggregate those different tests, and I'll explain what those different tests are. And so we call this star method. Then by the end, we create for each mask, we have association, like the ApoE enhancer and the LDL association, or ApoE promoter and LDL association. And so, but the question is, with many different functional weight, how can one combine the test with a different weight? So that's basically where the art is. So the traditional way, when we did this 10 years ago, and so basically one can develop this kind of burden test or stat test. So for the burden test, so basically you got this kind of individual association statistic. Basically you can think about the G transpose Y. And then this is your, for ApoE enhancer, you have 100 association statistics. You aggregate them, either using the linear combination or quadratic combination. And for the linear combination, so it is powerful when the effects are all in the same direction, because if the beta are all in the same direction, the linear combination will work out well. But when the effects are in different directions, and then the burden test will be a lousy choice, and then you want to do the quadratic test. And in that situation, basically this derived from the kernel machine framework, but turns out it has a very simple expression. And so this SCAD paper has been widely used in analyzing the whole genome sequencing data. And now, so this can be particularly useful, and when the variants are in different directions, especially in the non-coding regions, the enhancer and the promoter. Then the question is, ideally, what we want is for that weight function, if among the 100 variants, I know there are only 4 of them, beta not equal to 0, I want to give the weight 1 to those 4 variants and weight 0 for the other. And, but in reality, we don't know there are 4, and we don't know what they are. So therefore, what we do is we use this kind of Cauchy combination method and to combine correlated p-values. And so this is really a beautiful method when you deal with the test. We don't know there's a uniformly most powerful test, and you can try different tests to see which one give you the smallest p-value. If you use that, the smallest p-value to report as your final test, you're cheating because you look at the data multiple times. But the Cauchy combination test basically create a beautiful way to combine different correlated tests. And so basically it can be used to combine the SNP level p-value test, or it combines the p-values with, oh, this is even worse. Yay! Yay, thank you. Oh, so still one more. Oh, here we go. It works, thank you. Okay, and use the different weight. So let me show you how that Cauchy combination test works. So when you have a correlated test, and so traditional way to combine the correlated test is the Fisher combination test. And but this has a problem. And because when the tests are independent, and the Fisher combination test has basically 2B degree freedom test. But when the p-value are correlated, and this is not a chi-square test anymore. And so how can we account for that? So basically this Cauchy combination method create a beautiful way to combine the correlated p-value. So we don't like Cauchy. Generally, Cauchy doesn't have the mean. Cauchy doesn't have the variance. But it's really this kind of heavy tail distribution is really beautiful way to combine correlated p-values. And so the reason is, and no matter what is a p-value distribution look like, and the joint distribution look like, and the sum of Cauchy and still Cauchy in the tail, no matter what correlation you have. And so therefore, if you can see if we apply different tests to the same data set, all the p-value are correlated. But when we use a Cauchy to combine those p-values, still Cauchy, and the computation is super, super quick. So that makes things much, much easier to work with. And so this paper was first published in American Journal of Human Genetics and later in JASA. So that's basically how that works. So for example, if I want to test the ApoE enhancer, how it's associated with LDL, there are 100 variants, we can use Mendeleev frequency as a weight, run the scattered p-value, and then we do a Cauchy transformation, or you use the epigenetics APC as a weight, calculate the scattered p-value, do a Cauchy combination, or APC conservation as a weight, calculate the scattered p-value, do a Cauchy combination. We combine them using this Cauchy ACAT method, and computationally super, super fast is less than 1 second. And so we don't need to worry about the correlation at all. So that is very nice, and the accuracy is very good. And so for us, scalability is quickly important. So you can see for analyze the 500,000 people whole genome in UKB, and so for gene-centric analysis only take 1 hour. So that is very good. So here are the results for the top 30,000 people. You can see that for analyzing the different mask and LDL, the loss functional variance of PCSK9 is associated with LDL, and that's already a drug targeted, approved by FDA for PCSK9, and also NPC1L1 of missense variants is also associated with LDL, already a drug approved for that by FDA as well. So that's all encouraging. And the one thing is one can see that for the promoter and the enhancers, those are non-coding variants, the signals are weaker. So the one question is how can we improve the power of detecting enhancer and promoter region? And the answer is we have a postdoc of mine developed this cell star, and the idea is to integrate cell-specific functional annotation and using the single-cell data and the CRISPR and the perturbation data, and from the catalyst and or from the human cell atlas and or from the NIGR IGVF. So basically, you do the functional right, and then that will for the non-coding variants, that will provide a better discovery for the whole genome sequencing analysis. So this really creates a beautiful context for integrating different types of data. But when we do the data integration, we need to have a scientific problem in mind. So because you can have many different ways to do data integration, but how to integrate the data need to be driven by the scientific problem. Okay, so this is the last part of the talk. And so we did this work, and 10 years ago and also a couple years ago developed the STAR method. But we asked ourselves that 10 years later, is this the best we can do? And so here is that we are going to talk about how we can do the real variance association test using ensemble testing. So ensemble method has been widely used for prediction purpose, but our goal is not for prediction. Our goal is inference. And so we want to see how can we do ensemble inference. And so this paper was published, it will appear in GSSB, and led by my postdoc, former postdoc Xihong Lin. So here, I will present a simple context to understand the idea. So if you think about very simple regression, y equal to x beta. And I want to say, suppose the beta are all in the same direction, all positive. So the SNP in the loss functional variants, generally they're all productive or harmful for disease. I want to test the beta equal to 0. And so in order to do this test, so the traditional burden test is you assume all the beta, you aggregate totally the total dose level, and then take the beta out. So that basically you assume beta is the same. And then to do this test, this becomes 1 degree freedom test instead of 100 degree freedom test. And so basically the burden test, basically you aggregate the score statistic. A natural question is this the best we can do? So how can we develop scalable and powerful tests for testing beta equal to 0, this 100-dimensional beta? And so the challenge is the beta is multidimensional. The UMP test does not exist. And so for testing this multiple varied alternative tests, any test likelihood ratio test is not UMP test. It can lose power. So any test can perform poorly in a wide range of alternatives based on the Lemon lemma. So can we improve the power of this type of test? So the first question we ask ourselves, is this burden test the best one we can do if we know the signs are all in the same direction? The answer is no, because the burden test assumes all the beta to be the same, and but in reality, the beta can be in different directions, and even it can be in have different values. Some betas are big, some beta are small, and so the question is, how can we do better and to do this kind of a testing problem when we know that the likelihood ratio test is difficult to do in this kind of constrained problem, and it's not UMP test. And now let's think about for the prediction. And so for the prediction to do the learning, and so you can either find the best learner, and generally it is difficult and computationally expensive. The other way to do is ensemble. That is very effective in practice. And for our problem, we are not interested in prediction. We are interested in inference. And so to identify a single best test generally is mathematically and computationally challenging. So that motivates us to ensemble and to do the ensemble inference. So how can we do the ensemble inference? Let's start from ensemble prediction, ensemble learning. So ensemble learning is the classical machine learning method for prediction purpose. So the idea is to combine many weak base learners and to obtain a final strong learner. And so you have a bunch of weak learners. And then, so basically each weak learner is weak, but the final learner is strong. So basically this is similar to the Chinese idiom saying, the wisdom of the masses exceed the wisest individual. 三个臭皮匠顶个诸葛亮 for those who know Chinese. Okay, so now this is for prediction purpose. Okay, so a classical way and to do the ensemble learning is random forest. So basically in this situation, the base learners are trees, and those trees are all weak learners. And so then in order to build a strong learner, so you randomly select m variables. Generally, if you have 100 variables, you select 10. Take a square root of them. You want to make a tree independent. And then after you build you do that 1,000 times, and then you average those 1,000 trees. So basically this ensemble strategy is average, and base learner here is a tree. And so bagging is basically use all the variables, so that makes all the tree correlated. The variance is too big, so bagging generally doesn't work as well as the random forest. But this is for prediction purpose. I don't care about the prediction, I care about the inference. So now let's think about the burden test. Suppose I know the true signal direction, and those are the right lines. And so I want to ask myself, if I project the score statistic to some kind of random direction, which direction is likely to give me the best power? The answer is the true signal direction. So the burden test, if you think about the burden test basically is the sum of all the individual score statistics. So basically you project your score statistic to this 45-degree line. And even though I know the beta are positive, but I project to this 45-degree line. And so you can see if the true direction is not this 45-degree line, and then the burden direction will be a wrong direction, and those right direction, burden test will lose power, right? And so now, how can we find the better test than the burden test, given we don't know the true signal direction? So that is where this ensemble idea come in. So basically, first you build a weak-based test. So how to build a weak-based test? We basically do the random projection and project the score statistic to the random direction. Suppose all the betas are positive, and then I simulate 1,000 random direction in the first quadrant. How can I simulate random direction in the first quadrant? So basically, you simulate normal 0, 1, and you normalize it. That basically gives you the circle direction, the random direction. Now you project your score statistic to those 1,000 random direction. Many of those random directions are likely to be lousy direction. I don't care. As long as I have 1 or 2 directions which are closer to the true signal direction, I will be good, okay? But this also means that suppose you do this random projection of the score statistic 1,000 times, then you have 1,000 p-value. All those p-values are correlated. Many of those p-values are large because the directions are wrong, right? So averaging using the random forest strategy to do ensemble is a lousy idea, right? Because many of those directions are wrong, you're going to have a small number of directions to be right. So when we do ensemble, we are going to ensemble using ACAT. Use the Cauchy combination method, because this Cauchy combination method is very similar to the minimum p-value test. As long as 1 or 2 directions are right, that's going to give me that direction, and that gives me the best p-values. But it's better than the minimum p-test because I don't need to worry about the correlation. So that is really convenient. And we analyze the data using this ARIC whole genome sequencing data. So if we just look at the column on the right, and so we analyze lipid protein and neutroprophin. And so because we need to do this for a large number of time across the genome, so here we use allele frequency as a weight. So you can see if we don't ensemble, there are 6 discoveries for lipoprotein. But when we ensemble, we have 54, increase the discovery by 10 times. I did not have time to talk about ensemble scat, and one can also do that for ensemble scat. Then you can see when we ensemble it, and then increase the discovery by over 50%. So that is encouraging. To wrap up, and so this is the beautiful avocado, and so the beautiful Abu Dhabi. So first is it's important for the statistician to invest the time to do computer engineering and infrastructure building, and to build this kind of analytic ecosystem. And also it's important to make an impact in science. It's important to integrate the domain knowledge in statistical inference. And also it's useful for us, because statistician, we care about the inference. It's useful to think about from ensemble prediction or ensemble learning to ensemble inference. So if this is ensemble inference, it's a good thing. And finally, scalability is important, and especially when we deal with the cloud nowadays. Thank you.
It's very unfortunate that we had some technical difficulties, and I was just informed that the food is being prepared, but the restaurant is ready, so we have to move into the lunch so that we can enjoy the food at its prime. So we will resume the session that didn't end. We will resume, starting with Q&A with Xihong after lunch. And thank you.